Very good morning students. In our previous session, we have looked into the respiration, respiratory substrates, anaerobic respiration. We have dealt more clearly with respect to glycolysis, the energetics involved in glycolysis, what is the net ATP total energy, what is the net NADH, net ATP total energy released from glycolysis, and then we have also discussed about lactic acid fermentation, alcoholic fermentation. Right. So, in today's class we are going to discuss about aerobic respiration. Yes, what we are going to look into beta? Aerobic respiration. Aerobic respiration, where we are going to discuss more clearly about link reaction, which is also called as oxidative carboxylation followed by TCA cycle or Krebs cycle, TCA cycle followed by electron transport system, and then we are also going to look into amphibolic pathway and respiratory quotient So, all these segments are most important from this chapter beta. Definitely you will get at least 2 questions in your final NEET examination. So, stay focused for another 30 minutes, everything will be easy and clear to you. So, as we all know, glycolysis takes place in the cytoplasm, yes or no? If you consider this as a cell and this as cytoplasm, glycolysis occurs inside the cytoplasm, and at the end of glycolysis, what is produced? Glycolysis will break one glucose molecule to 2 molecules of pyruvic acid, yes or no? Glycolysis, in this process glucose is breaked to, or glucose is incompletely oxidized to, oxidized to 2 molecules of pyruvic acid. These 2 pyruvic acid molecules will enter directly into the Krebs cycle. Okay, these 2 molecules of pyruvic acid will directly enter into the mitochondrial matrix. They will enter into mitochondrial matrix. Inside the mitochondrial matrix, 1 molecule of pyruvic acid linked with 1 molecule of pyruvic acid along with coenzyme A and 1 molecule of NAD plus, these 2 coenzymes, in the presence of the enzyme pyruvate dehydrogenase, it will release out 1 molecule of acetyl CoA followed by 1 molecule of NADH, followed by energy release of 1 molecule of NADH, and 1 carbon dioxide is released out. Pyruvic acid is a 3 carbon compound, acetyl choline is a 2 carbon compound. So, this step wherein pyruvic acid is converting to 2 carbon, that is acetyl CoA, is called as link reaction beta. This is called as link reaction. Or transition reaction or transition step or reaction. Okay, this step is called as what ma? Link reaction or transition reaction, or it is also called as oxidative decarboxylation. Clear? So, yes, this link reaction or oxidative decarboxylation, yes, just a second. link reaction, oxidative decarboxylation or transition reaction occurs in the mitochondrial matrix, wherein one molecule of pyruvic acid is converted to one molecule of acetyl-CoA, releasing out carbon dioxide and NADH. Clear? So, yes, if we then look into the acetyl-CoA, acetyl-CoA will further enter into TCA cycle, which is nothing but tricarboxylic acid cycle, which takes place again in the mitochondrial matrix itself. TCA cycle is also called as Krebs cycle. TCA cycle is also called as Krebs cycle, named after the scientist Hans Krebs. Okay. So, yes, TCA cycle, Krebs cycle or citric acid cycle, okay, or citric acid cycle as the first product formed, first stable product formed in this reaction is citrate. So, it is called as citrate cycle or citric acid cycle as well. If we clearly look into the steps of TCA cycle, here acetyl-CoA along with oxaloacetic acid. Oxaloacetic acid is a 4-carbon compound, acetyl-CoA is a 2-carbon compound. They get condensed to form the first molecule, that is citrate. Okay, this is a condensation reaction. And they will form citrate by the enzyme citrate synthase, and then citrate is turned to isocitrate by the enzyme cis-aconitase. Okay, by the enzyme cis-aconitase, isocitrate is formed. Citrate is 6-carbon, isocitrate is also 6-carbon compound. Then isocitrate along with the cofactor or along with the NAD plus, it is turning to NADH by the enzyme isocitrate dehydrogenase. So, this is the step where one NADH is released. If we are considering one acetyl-CoA, one Krebs cycle I am talking about, in one Krebs cycle, at isocitrate one NADH is released by the enzyme isocitrate dehydrogenase, and then 5-carbon compound, that is alpha ketoglutarate, is formed. 6-carbon compound isocitrate is forming out alpha ketoglutarate, that is 5-carbon compound. And here again by utilizing NAD plus, NADH is formed. Okay, alpha ketoglutarate is again utilizing NAD plus, and it is releasing out the energy rich molecule, that is NADH, and forming a 4-carbon compound, that is succinyl-CoA, second decarboxylation reaction, which is catalyzed by the enzyme alpha ketoglutarate dehydrogenase. Succinyl coenzyme A is forming out succinate by utilizing GDP. This is said to be substrate level phosphorylation, because GDP is utilizing phosphate group from succinyl CoA and forming out GTP. ATP can also be formed. It can be ADP to GTP or GDP to GTP. Okay, it can be from GDP to GTP or ADP to ATP as well. Okay, wherein succinyl coenzyme A is forming out succinate. Okay, succinyl coenzyme A is forming out succinate, which is, this step is what ma, substrate level phosphorylation, where it is releasing one molecule of ATP or GTP, and this is catalyzed by the enzyme succinyl coenzyme A synthase, succinyl CoA synthase. Next, succinate, which is 4-carbon compound, is forming out 4-carbon compound, that is fumarate. In this step, this is catalyzed by the enzyme succinic dehydrogenase. All the enzymes that are involved in Krebs cycle are present in the mitochondrial matrix, but this enzyme, that is succinic dehydrogenase, is found attached to the inner mitochondrial membrane. And this is found attached to the inner mitochondrial membrane. Okay, just a second. Yeah, if you consider this as mitochondria and this is the outer mitochondrial membrane, inner mitochondrial membrane, and this is mitochondrial matrix, and this is intermembrane space. Okay, here Here succinate dehydrogenase enzyme is found attached to inner mitochondrial membrane towards the mitochondrial matrix. Clear? So, here succinate to fumarate by releasing out one molecule of FADH2, and then fumarate is turned to malate. Malate is forming out oxaloacetate, which is 4 carbon, okay, by the enzyme malate dehydrogenase, and this step is also releasing out 1 NADH. So, this is all about TCA cycle. In this, what we have to remember the most? What we have to remember the most? If you look into acetyl coenzyme A, which is formed at the end of link reaction, one acetyl coenzyme A enters one Krebs cycle. First, 2 carbon compound acetyl-CoA is reacting with 4 carbon compound oxaloacetate, forming out 6 carbon compound, that is citrate. Citrate is again forming out isocitrate by the enzyme cis-aconitase. Isocitrate is formed to be a 5 carbon compound, that is alpha ketoglutarate, by releasing out one molecule of NADH, and this is catalyzed by the enzyme isocitrate dehydrogenase. In this step, one NADH and one carbon dioxide is released out. Then 5 carbon compound alpha ketoglutarate will form succinyl coenzyme A, which is 4 carbon compound, and this also releases one NADH and one carbon dioxide. Succinyl CoA forms out succinate by forming one GTP or one ATP molecule, which is said to be substrate level phosphorylation. Next, succinate to fumarate, which is 4 carbon compound, catalyzed by the enzyme succinic dehydrogenase, and here also one FADH molecule is released. Fumarate to malate, where 4 carbon compound is formed by the enzyme fumarase. Malate to oxaloacetate, which is the last step of Krebs cycle, where again one NADH molecule is released. So, here if you see the energetics involved in Krebs cycle, high amount of energy in aerobic respiration is released during TCA cycle itself. So, here at 3 places for one acetyl CoA if we consider, or one molecule of pyruvic acid, or one molecule of pyruvic acid if we consider, we have to consider the link reaction also. Yeah. So, here one molecule of pyruvic acid will form out one NADH2 at the end of link reaction. During link reaction, one NADH is formed. At link reaction. And here from isocitrate to alpha ketoglutarate in Krebs cycle, in TCA cycle, from here to here, isocitrate to alpha ketoglutarate, one NADH is released, alpha ketoglutarate to succinyl coenzyme A, one NADH, and here malate to oxaloacetate, one NADH. So, total 3 NADH are formed. How many ma? 3 NADH. If he is asking you from one acetyl CoA, then you have to answer 3 NADH. Okay? And one molecule of FADH, from succinate to fumarate, it is 1 FADH2, and in Krebs cycle one ATP is also released. In Krebs cycle. This is for one TCA cycle. In one Krebs cycle or one TCA cycle, 3 NADH, 1 FADH2 and 1 ATP is released. In link reaction for one pyruvic acid, 1 NADH is released. Clear? Now, if we calculate the complete energy that is released during aerobic respiration after the discussion of the energy sheet, then we will go for the discussion of electron transport system. In complete aerobic respiration wherein complete oxidation of glucose happens, and in this Krebs cycle if you see, there is a stepwise removal of hydrogen ions, okay, there is a stepwise removal of hydrogen ions and electrons, and there is stepwise oxidation process. and this aerobic respiration will release out huge amount of energy. So yes, if we look into this as a cell, and in glycolysis of course glucose is partially oxidized or incompletely oxidized to two molecules of pyruvic acid by releasing out 2 ATP and 2 NADH as the energy, okay, by releasing out 2 ATP and 2 NADH. This pyruvic acid, 2 molecules of pyruvic acid will enter into the mitochondria. It will enter into what ma? Mitochondria, into the mitochondrial matrix. So inside this mitochondrial matrix, pyruvic acid is first turned to acetyl-CoA. Pyruvic acid is turned to acetyl-CoA by releasing out how many molecules of NADH ma? We are calculating for 1 glucose, no? So for 1 glucose it is 2 molecules of pyruvic acid, 2 molecules of acetyl-CoA are formed, and it is 2 NADH. It is 2 NADH, and these 2 molecules of acetyl-CoA, after entering the TCA cycle, it forms out 6 NADH, 2 FADH2, 2 FADH2 and 2 ATP molecules. 2 FADH2 and 2 ATP molecules. This is the energy chart of aerobic respiration. So if you look into 2 ATP and 2 NADH, in glycolysis it is, it is 2 ATP and 2 NADH. 2 NADH, 1 NADH is equal to 3 ATP, right? So 2 into 3, how much ma? 6 ATP molecules, okay. Of the 6 ATP molecules, there is a shuttle system which is activated in this inner mitochondrial membrane. This shuttle system is called as glyceraldehyde 3-phosphate shuttle system, okay. This shuttle system is called as what beta? Glyceraldehyde 3-phosphate shuttle system. To pump 1 NADH energy molecule to the mitochondrial matrix, it will utilize 1 ATP. So to pump 2 NADH molecules, into the mitochondrial matrix, of course it will utilize glyceraldehyde 3-phosphate shuttle system will utilize 2 ATP. So in eukaryotic cells, NADH, to transfer the energy from this NADH into the mitochondrial matrix, 2 ATP molecules are utilized. So 6 minus 2 is utilized by glyceraldehyde 3-phosphate shuttle system. It is 4 ATP. How much ma? It is 4 ATP. And this is the energy of glycolysis in aerobic respiration. And next is link reaction. In link reaction, how much energy is released? Of course, it is 2 NADH. To convert it into ATP molecules, 2 into 3, 6 ATP. Okay. Next, third is after link reaction, it is TCA cycle, right? TCA cycle. In TCA cycle, of course, we get 6 molecules of NADH, right? 6 NADH, because we are considering for 1 glucose molecules. 1 glucose molecule, it is 6 NADH, 2 FADH2 and 2 GTP or ATP. You can consider anything. Okay. So here 6 into 3, it is 18 ATP molecules, and 2 into 2, 1 FADH2 is equal to 2 ATP, right? So here 2 FADH2 means here 4 ATP, and then 2 GTP or 2 ATP molecules. Fine. If you calculate the complete ATP molecules, which are formed during aerobic respiration in a eukaryotic cell. It is 2 ATP plus 4 plus 6 plus 18 plus 4 plus 2. On total it is 36 ATP. This much of energy is released in a cell, this which is called as cellular energy utilized for all the metabolic activities inside the cell. So, this energy is released at the end of aerobic respiration. How much energy? 36 ATP in a eukaryotic cell. Okay, without shuttle system, so it is 38 ATP, but in a eukaryotic cell, what is the amount of energy that is released at the end of aerobic respiration? If this is the question, then you have to answer it is 36 ATP. Whenever the questions are given, beta, you have to show keen interest, keen focus, whether he is asking you for one glucose molecule or for one pyruvic acid or for one acetyl CoA or two acetyl CoA. You have to look into the substrate molecule or from which step he is asking the question. Based on that, you have to answer the energetics of the aerobic respiration. Till now, if you have looked into glycolysis, link reaction and Krebs cycle, nowhere you have observed the release of oxygen and a very little ATP is formed, because in Krebs cycle if you see, energy rich molecules like NADH, FADH2 and in glycolysis also NADH is formed, but this 36 ATP is not formed in these two cycles, both in glycolysis, TCA, link reaction. ATP molecules are formed and oxygen is formed at the final step of aerobic respiration. That final step of aerobic respiration is called as electron transport system. What is it called as, beta? Electron transport system. Electron transport system. Yes. If you look into here, electron transport system, and once again if we consider the mitochondria, this which is the outer mitochondrial membrane and this is the inner mitochondrial membrane, this is mitochondrial matrix. Inside this this is outer mitochondrial membrane, inner mitochondrial membrane which is also called as cristae, and this is intermembrane space. Clear now? So, here inside the mitochondrial matrix all the energy rich molecules are present. We have NADH combining link reaction and TCA cycle. How many NADH we have, ma? In mitochondrial matrix we have 8 NADH, 2 FADH2 and 2 ATP. Okay. This is the amount of energy that is present in the mitochondrial matrix. Now, all this complex form of energy have to be converted to simplest form of energy, that is ATP, which is called as cellular energy. This is done through electron transport system. So, here in the mitochondrial matrix, whichever energy rich complexes are present, they are forming a channels. You know how many complexes? 14 protein channels, 4 protein channels or complexes we say, or complexes. These are operated, 4 protein channels or complexes are operated from mitochondrial matrix to intermembrane space. Complex 1 is called as NADH dehydrogenase. It is called as NADH dehydrogenase. Okay. Complex 2 is called as succinate dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is called as cytochrome BC complex. It is called as cytochrome BC complex. Complex 4 is called as cytochrome C oxidase. Complex 4 is called as cytochrome C oxidase. Clear? So, if you look into the complex 1, Complex 1 is a transmembrane channel which is operated from mitochondrial matrix to intermembrane space. It is from mitochondrial matrix to intermembrane space beta. So, here if you see the NADH molecules, NADH molecule is cleaved to NAD plus, okay, NAD plus by the enzyme NADH dehydrogenase. So, both protons and electrons are released. Protons 4H plus are immediately pumped to the intermembrane space, taking out 2 electrons. 2 electrons are being transferred through protein molecules which are present inside the complex 1. Those are FMN and ferrous sulfur complex. Okay. NADH molecule is releasing out NAD plus H plus electrons plus 4H plus 2 electrons. Okay. This 4H plus molecules, 4 protons are pumping out to the intermembrane space, and these 2 electrons are being pumped to the protein molecules which are present in this complex 1. Those are FMN complex and ferrous sulfur molecule. Okay. FMN and ferrous sulfur molecule, and then mobile electron carrier, that is ubiquinone. Okay, we will discuss about this. As of now, you remember that from complex 1 NADH dehydrogenase, NADH plus H plus is braked to NAD plus 4 protons and 2 electrons. These 4 protons are pumped to the intermembrane space and 2 electrons are carried through the protein complex by protein molecules FMN and ferrous sulfur complex. Okay. If you look into the complex 2 is called as succinate dehydrogenase. Complex 2 is called as what ma? Succinate dehydrogenase, where 4 carbon succinate is converted to 4 carbon fumarate. Okay. I already said you succinate dehydrogenase is found attached to the inner membrane, inner mitochondrial membrane. So, yes, here the protein, both electrons and protons are transferred through the protein channel only, because this channel is not transmembrane protein channel. This is embedded in the inner mitochondrial membrane itself. So, here both electrons and protons are submitted to FAD plus. Both electrons and protons are submitted to FAD and then ferrous sulfur complex. Okay. Both electrons and protons are submitted to FAD and ferrous sulfur complex, and then the protons and electrons are from here complex 2 and complex 1 electrons are submitted to ubiquinone. Ubiquinone, which is called as mobile electron carrier, mobile electron carrier, because this is carrying electron between 2 successive complexes. Okay. And this turns out to be ubiquinone along with hydrogen, and here it pumps out the protein molecules from complex 3. Okay. Both H plus ions and electrons are transferred through this protein molecules submitted to ubiquinone, and from here it is given to the intermembrane space. And electrons, protons are submitted to the intermembrane space by ubiquinone, and electrons are given out to cytochrome B protein which is present in the complex 3. From cytochrome B to ferrous sulfur complex and cytochrome C. Cytochrome C is again a mobile electron carrier. Cytochrome C is again what beta? Mobile electron carrier. Okay. This mobile electron carrier is submitting out the electrons to copper molecule. If you look into, in all the 3 complexes, the molecule which is present is ferrous sulfur complex and ferrous sulfur complex. In complex 4, the molecule which is present is copper ions. The ions which are present are copper ions and cytochrome A and A3. Here the electrons are transferred, 2 electrons. Okay. What is the electron channel? How much electrons are the protein molecules transferring? 2 electrons, and ultimately the electrons are accepted by oxygen atom. The question, very important, what is the ultimate electron acceptor or the final electron acceptor in the electron transport system inside mitochondria is oxygen. Accepting 2 electrons and protons which are present in the matrix, it forms out water molecule. It forms out what ma? Water molecule, accepting both electrons from complex 4 and protons which are already present in the mitochondrial matrix, it forms out water molecule. Okay. And then this is all about complex 4. once again I will give you a quick recall about all these complexes on how electrons and protons are transferring. First complex, which is called as NADH dehydrogenase, wherein NADH plus H plus is formed out to be NAD plus 4 protons and 2 electrons. 4 protons are immediately pumped to the intermembrane space and 2 electrons are carried through the protein molecules which are present in complex 1. Those are FMN and FES. Do remember, students. Second complex 2 is not a transmembrane protein channel. This is embedded inside the inner mitochondrial membrane, which is called as succinate dehydrogenase. Here the electrons are pumped to FAD and ferrous sulfur complex, okay, FAD and ferrous sulfur complex, and here electrons are given to a mobile electron carrier ubiquinone. From complex 2 though, both electrons and protons are given to ubiquinone, and this is pumped to the intermembrane space, 4 protons, and electrons are carried through cytochrome B, ferrous sulfur complex of complex 3, and then to a mobile electron carrier, that is cytochrome C. Cytochrome C is a mobile electron carrier which is present outside of the inner mitochondrial membrane in intermembrane space, okay. The mobile electron carrier between 2 channels, that is complex 1 to complex 3 and complex 2 to complex 3 is ubiquinone, and the mobile electron carrier between complex 3 and complex 4, this is present towards the outside of the inner mitochondrial membrane, that is cytochrome C. This is giving out the electrons to copper molecules and then cytochrome A, and the ultimate electron acceptor is oxygen molecule, okay. This oxygen molecule along with the protons which is present in the mitochondrial matrix and electrons, this forms out to be water molecule. So, if you clearly look into whatever H plus are released out as a result of oxidation, be it the oxidation of NADH or FADH2, whatever H plus molecules are released are pumped out to the intermembrane space. 4 H plus molecules from complex 1, 4 from complex 3 and 2 from complex 2, okay. After forming of water molecule, 2 H plus are being pumped from complex 4 to intermembrane space. This proton molecules are released into the intermembrane space, and whichever little H plus molecules are present in the matrix are forming out to be water molecule. As a result, proton gradient is generated from intermembrane space to mitochondrial matrix, whereby ATP synthase complex is activated, which is called as F0 F1 complex. This is called as what, beta? F0 F1 complex, where F0 is a protein channel which is embedded inside the mitochondrial matrix. F1 is facing towards the F0 is a protein channel, F0 is a protein channel which is embedded in the inner mitochondrial membrane. F1 is facing towards the mitochondrial matrix, okay. This is very important, you have to remember. F0 is inside the inner mitochondrial membrane, F1 is facing towards the mitochondrial matrix. So, like this it is being operated. This is F0 and this is F1 towards the mitochondrial matrix. At a single stretch, this releases out 4 H plus molecules, which gives the sufficient energy for phosphorylation of ADP plus inorganic phosphate, whereby it forms the ATP molecule. This is called as oxidative phosphorylation. Oxidative phosphorylation. In photosynthesis, we will learn it as non-cyclic or cyclic photophosphorylation, okay. In photosynthesis, we learn it as photophosphorylation. Here in mitochondria, what is happening? Oxidative phosphorylation, where in a single pump it releases out 4 protons which forms out 1 ATP molecule, okay. Combining ADP and inorganic phosphate which is in mitochondrial matrix, it forms ATP molecule. Clear? This is all about electron transport system. What you have to remember from this? All the 4 protein complexes and the protein molecules which are involved in each complex that are carrying out 2 electrons, and what are mobile electron carriers here, and which protein complexes are transmembrane and which is embedded inside the inner mitochondrial membrane, and At final, that is, we will also call it as complex 5, which is nothing but ATP synthase. F0 is embedded inside the inner mitochondrial membrane and F1 is towards the mitochondrial matrix. Clear? This is what you have to remember, okay, and this finally forms out to be ATP molecule, which is called as oxidative phosphorylation. So now at the end of electron transport system, you can see out oxygen molecule, water molecule and ATP molecules are released out. So as we discussed, the energy that is released from aerobic respiration is in eukaryotic cells. In eukaryotic cells, the energy released out in the aerobic respiration is 36 ATP molecules, wherein 2 molecules are utilized by what beta? Glyceraldehyde 3-phosphate shuttle system, and this is about electron transport system. Now we are going to look into amphibolic pathway, okay. Respiration is also called as amphibolic pathway, amphibolic pathway, wherein mostly the favorite respiratory substrate is glucose only, okay. Glucose is mostly used as respiratory substrate, but then proteins and fats are also acting as respiratory substrate sometimes, okay. Proteins are being catalyzed to, or proteins are breaked to amino acids, which will later convert into pyruvic acid. Fats are also breaked to fatty acids and glycerol, wherein glycerol converts to DHAP, that is dihydroxy acetone phosphate, and fatty acids to acetyl CoA. So whichever might be the respiratory substrate, TCA cycle is common for all the respiratory substrates, okay. TCA cycle or Krebs cycle is common for all the respiratory substrates, be it acetyl CoA or succinyl CoA, whatever it might be, or alpha ketoglutarate also. These act as intermediates. These respiratory intermediates will act, or they play major role in biosynthesis of many other molecules like secondary metabolites, like secondary metabolites like carotenoids, terpenes, okay, and many other biosynthetic pathways. All the respiratory intermediates play major role in the biosynthesis of many other processes, also many other secondary metabolites, like in those biosynthesis processes, these respiratory intermediates will play major role, like succinyl CoA or acetyl CoA or alpha ketoglutarate acid. And here in amphibolic pathway, what you have to know beta, whatever might be the respiratory substrate, if it is fats, it gets converted to DHAP and then it enters to the Krebs cycle. If it is proteins is acting as respiratory substrate, it turns to pyruvic acid and then it enters the Krebs cycle, okay. As such, so Krebs cycle is common for all the respiratory substrates. Last segment of this chapter is respiratory quotient. What is it, ma? Respiratory quotient. It is nothing but the volume of carbon dioxide evolved by volume of oxygen consumed during respiration, okay. What is respiration quotient? Amount of carbon dioxide evolved by the amount of oxygen consumed. If glucose is the respiratory substrate, if glucose is the respiratory substrate, wherein 6 carbon dioxide molecules are evolved, 6 carbon dioxide molecules are evolved and 6 oxygen molecules are utilized. So 1 is the RQ value if glucose is the respiratory quotient. If glucose is the respiratory substrate, RQ value will be is equal to 1, okay. If tripalmitin or fats, okay, fats are acting as respiratory substrate, then amount of carbon dioxide evolved will be more than will be less than the amount of oxygen utilized. More oxygen needs to be utilized for the release of carbon dioxide molecules. So in this case it will be less than 1. For fats it is RQ value is 0.7. Same in the case of proteins. Proteins also it is less than 1, but RQ value in proteins is 0.9. Okay. If you consider alcoholic fermentation or anaerobic respiration, okay, wherein the carbon dioxide that is evolved at the end of alcoholic fermentation is what, ma? Ethanol plus 2 carbon dioxide, right? So, 2 carbon dioxide, and there is no oxygen utilization only, there is no oxygen consumed during anaerobic respiration, because anaerobic respiration takes place in the absence of oxygen. 2 carbon dioxide with 0 oxygen, something by 0 is always infinity. So, RQ value is infinity for anaerobic or alcoholic fermentation. So, glucose, fats, proteins and alcoholic fermentation. Next comes the organic acids. Oxidation of this organic acids, if you consider the RQ value, it will be greater than 1, okay, it will be greater than 1. If you consider oxalic acid, it is 4, okay, oxalic acid RQ value is 4. Acetic acid, RQ value is what, ma? 1.3 as such. So, these are the RQ values, of different respiratory substrates, okay. So, this is not acetic acid, this is malic acid, yeah, malic acid, fine. So, these are different RQ values for different respiratory substrates, which is most important to remember. So, what all which are required for this chapter, we have completed the chapter respiration in plants. In the coming up sessions, we are going to deal with plant growth and development. So, stay tuned. Thank you.